Good morning, dear students of class 12 science. So welcome to biology class. We are dealing with the chapter biotechnology and its application. So this is the third class of this chapter. So in today's class we will start with the topic application of biotechnology in the field of agriculture. Okay, so all of you have heard about the term green revolutions, right? Where there was mainly a movement, let's say, which was mainly done to increase the productivity of the agricultural crops. So this particular technique was mainly used to increase the productions of the crop. Clear? So green revolutions, we have said that it was mainly used for the productions of those crops, but it was not enough to feed the rapidly increasing human populations. Though these revolutions, it increased the productivity of the crops, but it was not able to feed the rapidly increasing human population. Clear? So the increased production of food grains had been due to the use of new and improved varieties of crops, the judicious use of agrochemicals, okay, and better irrigation and better management practices. So these were the few regions where there was rapid increase of food grains during the green revolutions. Now, at present time, conventionally, the genetic variation is necessary for the crop improvement. Okay, so we have to go for genetic variations within the crop, and this can be only done through the crossbreeding. It can be done by the help of hybridizations. It can be done by the help of mutagenesis and polyploidy. But this particular processes, they are very time consuming and they are also not perfect. Okay, but now in the present time, the use of chemical fertilizers, the use of pesticides are totally banned in most of the states. So that's why the use of agrochemicals, they have been replaced by the use of biofertilizers, and they have been replaced by the use of biopesticides. So these biofertilizers, biopesticides, we have already learned in our previous units, to try to recall it back. Okay, so growing crops, fruit, vegetables using biofertilizers and biopesticides is called organic farming or the organic agriculture. So development of genetically modified crops by the help of genetic engineering can overcome all these problems. Okay, so these conventional methods of increased crop productions have the following limitations. What are those limitations or drawbacks? Poor farmers or developing countries cannot afford expensive agrochemicals. That is fine, they are not able to buy those heavy expensive let's say chemical fertilizers. They are not able to buy those pesticides. Number two is the indiscriminate use of agrochemicals. That means those chemical fertilizers, those pesticides, those insecticides, they will decrease the fertility of the soil. Clear? And not only that, they will decrease the fertility of the soil, but they also will have an adverse effect on the health of the human being, because we are at the top of the food chain. So whenever we are applying those chemical fertilizers on the agricultural fields, on the producer level, the concentration of those chemicals will keep on increasing throughout the food chain, throughout the trophic level, and ultimately we are the one who are at the top of the food chains will be affected by this effect. And number three limitation is that the existing varieties of crops further increase in the yield cannot be expected. So these are the few limitations of the use of agrochemicals, we can say. Okay, now what do you mean by genetically modified plants, GM plants, or the transgenic plants? Look at definitions. Okay, the plant obtained by the transfer of gene or the genes from some related organisms through genetic engineering are called transgenic plant. Or we can also say that transgenic plants are those plants whose genes has been altered or whose genes has been modified. Okay, so what we are doing is that in those plants we are transferring a gene or the genes from the related species, and in that case those plants will be modified, their genetic compositions will be altered. So those plants whose genes has been altered or has been modified are known as genetically modified plants or the transgenic plants. So the first transgenic plants was produced in 1983 when a new variety of tobacco expressing cannabinoid resistance was produced. Okay, then after that we have got lots of varieties of transgenic plants which has been produced. So Flavor tomato was the first variety tomato which was there to reach the market. Okay, so remember the term flavors tomato, which was the first genetically modified tomato, was the first variety to reach the market. Now such varieties are popularly termed as designer crops. Remember the term, such varieties, they are popularly known as designer crops because they are developed to meet the certain specified requirements. Okay, why are we making those genetically modified plants? Because we have got certain requirements. Okay, and to fulfill those requirements, in order to meet those requirements, we are creating the transgenic plants or the transgenic crops. So flavors tomato was the first variety to reach the market, which was developed by the help of genetic engineering, and it was mainly known as designer crops because they were developed to meet the certain specific requirements. Clear? So we will talk about the objectives of developing the GM crops. Why we are developing the genetically modified plants or the genetically modified crops. The first one is pest resistance. Okay, so farmer, they used to use lots of agrochemicals, they used to use lots of insecticides, pesticides to protect their plants from the pest infestations, from the pest infections. But we have told that the use of those chemicals will be very harmful, not only to the soil, that it will not decrease only the soil fertility, but it will also hamper the health of the human beings. So the use of those agrochemicals must be stopped. Okay, it must be replaced by the use of those biopesticides and biofertilizers. Okay, so to eliminate the applications of chemical insecticides and the pesticides, pest resistant genetically modified crops are being developed. Okay, so few examples of the pest resistant crops which has been developed are BT cotton. Afterwards we learned about BT cotton, what is BT means over here. Okay, so BT cotton is one of the genetically developed pest resistant cotton varieties, which is called BT cotton. It resistance to the insect larvae. And we have got nematode resistance varieties of potato, tomato and brinjal. So these are the few examples of the plants or the crops which are being resistant to the pest and which have been developed by the help of genetic engineering. Then number 2 we have got is herbicide or weedicide tolerance. Okay, so manual removal of weeds from the crop field is both expensive as well as time consuming for the farmer. This is true, and therefore the farmer also used to spray large quantities of herbicides in order to remove the unwanted herbs from the agricultural fields, which will pollute the environment also. So genetically modified plants of cotton, maize, tobacco, soybean with herbicide tolerance have been developed. So we are developing the plants which are tolerant to the herbicides, and they are able to biodegrade herbicides also. So we are developing these particular crops or the plants by the help of genetic engineering. Then number 3 we have got tolerance of abiotic stresses. So those particular genetically modified crops, they are able to tolerate the frost, they can grow. Okay, they can tolerate frost, cold, heat, drought, salinity or heavy metal toxicity, with the normal crop cannot resist. Number 4 is disease resistance. So genetically modified plants, they are resistant to infectious disease also. Okay, and they are being developed to save our crops and forests from the fungal, bacterial and the viral attack. For example, look over there, we have got the viral resistance varieties of tobacco, rice, papaya, potato and tomato. Okay, so this has been developed already, which has been resistant to the viral attack. Then transgenic crops with increased resistance to fungal pathogens have also been developed. So these have the transgenes for PR proteins or the enzyme chitinase to destroy the fungal cell wall. So those plants, they have got the chitinase enzymes or the PR proteins which will usually then destroy the fungal cell wall, and those plants will remain unaffected from the fungus. Okay, so whenever any plant is being infected by the fungus, then this enzyme chitinase or the PR protein, the gene for the PR protein or less for enzyme chitinase, will destroy the fungal cell wall, and the fungus will not be able to survive, and that plant will be free from the fungal attack. Number 5 is reduction in post-harvest losses. So fruits and vegetables with long shelf life are being developed. That means after collection, okay, we used to store the food, but during storage, lots of foods used to be wasted. They start rotting. So what I mean is that in this case, by developing GM crops, we are increasing the shelf life of the plants, of the crops, so that they can remain fresh for the longer period of time. One such example is flavor tomato. Just we have been mentioned that one example of GM crops. This flavor tomato has got a long shelf life period. That means it got long post harvesting period. Okay. Next one is the enhanced nutritional value of food. So malnutrition is common in third world countries where people rely on a single crop. Okay. So therefore, in order to overcome the malnutrition, we are developing lots of plants, lots of crops which can overcome this such nutritional deficiencies. So one example is the golden rice. Remember carefully, golden rice is very, very important because this golden rice is enriched with vitamin A and potato rich and protein rich potato. Generally they will ask the questions, what is significance of golden rice? So golden rice, they are enriched with vitamin A. Okay, so this has been developed through genetic engineering in order to overcome the problem of nutritional deficiency or the malnutrition. Then we have got next one is mineral utilization, where crop plants have been modified for increased efficiency of mineral utilizations. And this conserves the soil fertility and reduces excessive addition of fertilizers, because once we are using the agrochemicals, they used to decrease the soil fertility. Okay, they used to decrease the soil fertility and in return they used to affect the human health. But over here, by developing the GM crops, genetically modified crops or the genetically modified plants, there will be increased efficiency of mineral utilization by the soil and should also conserve the soil fertility. Okay, so there will be no loss of soil fertility by the development of genetically modified crops. Clear? Next one is pharmaceuticals, where we are using the transgenic plants which are mainly used to produce the different types of pharmaceutical drugs like insulin. We are developing vitamin A, we are developing interferon, the blood clotting factors, immunoglobulin and the vaccines. So these are being mainly done by the help of genetic engineering process. Okay, so cultivation of transgenic plants for pharmaceuticals is called farming or molecular farming. Then we have got few edible vaccines, point number 9, where scientists are working on edible vaccines like in tomatoes and the potatoes for the several indirect pathogens, which can be fed to animals or humans for immunizations or for vaccinations. Then we have got number 10 is the commercial products, where plants are being engineered to yield higher amount of biopolymers and commercial products. Okay, like nylon, biofuel or the biodiesels, which are mainly being employed, which have been discovered mainly for the commercial purposes. So these are the few objectives of developing the GM plants or the GM crops. And so we will see the few GM plants or the GM crops which has been developed. Okay, so go through the page number 625, table number 12.3. You will see the name of the few transgenic plants. Okay, the first one is cotton, and the transgenic cotton is known as BT cotton. Okay, and transgenes beta-2 toxin gene cry2AB, this we will learn afterwards. Just go to the useful characteristics. They are resistant to bollworm and insect larvae. Similarly, rice, the golden rice, it is rich in vitamin A. Rice grains are golden yellow in color and they are also rich in vitamin A. Tomato, it was flavor tomato, which are developed. Characteristics, useful one, they have got long post harvesting time. Okay, so delayed ripening, and they have got improved flavor and insect resistance property. Then we have got potato, the name is new leaf. Useful character is that they are insect and virus resistance, and starch content is also increased by 20 to 40%. So here you can see the development of the few transgenic plants along with the useful characteristics. Wheat, Basia. Useful characters, their resistance against the herbicides PPT. Then we have got mature corn, and name is yield guard BT cry1AC gene, and the useful characteristics of this mature corn is that they are pest and insect resistance. And last one is tobacco, they are resistant against the pest. So these are the few examples of the transgenic plants which has been developed by genetic engineering along with their useful characteristics. Clear? So go through it. Go through table number 12.3. Now we will see the first one, that is BT cotton, because we are saying that we have got the BT cotton which has been developed by the help of genetic engineering and which are resistant to bollworm and insect larvae. So how these varieties has been created or has been developed by the help of genetic engineering? So BT cotton is mainly a transgenic variety of cotton. Okay, it's mainly a transgenic plant. So genes has been altered by the transfer of gene or the genes from the related species. So BT cotton is mainly a transgenic plant or the transgenic cotton, and it's mainly been engineered by introducing a gene from Bacillus thuringiensis. This is very important. Okay, so over here, this particular curtain has been engineered, has been produced by engineering a gene from the bacterium called Bacillus thuringiensis. And this bacterium, they used to produce the insecticidal protein, which is toxic in nature, and this insecticidal protein used to kill the larvae of the insects which belongs to the order Lepidoptera, like armyworm, tobacco bollworm, or of the order Coleoptera, like beetles, Ambidiptera, flies and mosquitoes. Remember one more thing, that BT curtain is mainly a transgenic crop which has been developed by the genetic engineering technology. The gene has been introduced from the bacterium Bacillus thuringiensis. So therefore in the name BT curtain, the BT stands for Bacillus thuringiensis. Clear? So I can remember this one. The term BT, this BT stands for the name of the bacterium Bacillus thuringiensis. Okay, because this bacterium, they used to produce the insecticidal protein, and that insecticidal protein is toxic in nature and is mainly used to kill the larvae of the insects. Okay, so let's go in details about this one. So there are a few strains or the subtypes of the bacterium Bacillus thuringiensis. They used to produce the endotoxin. Endotoxin means they are produced within the body. Okay, so they used to produce the endotoxin proteins that used to kill the insect. And these proteins, they used to be in the form of crystals. They are deposited in the form of spores, and therefore they are called crystalline proteins or the cry proteins. And these cry proteins, they are produced by the help of the gene called the cry gene. Clear? So the gene for the toxic cry protein is obtained from the soil bacteria Bacillus thuringiensis. This gene is known as BT gene also. Since this particular gene is obtained from the Bacillus thuringiensis, so it is called BT gene, and the protein that is encoded by this BT gene is known as BT protein. Okay, so this particular BT protein is mainly, which is toxic in nature, is endotoxin. It is produced within the body of the bacterium, that is Bacillus thuringiensis. But this particular cry protein, it is non-toxic to the bacterium. Why? Because within the body of bacterium, this cry protein, which is toxic in nature, will be in inactive form. One more time saying that this BT Bacillus thuringiensis, which is producing the cry protein, which is toxic in nature, is not affected by this toxic protein itself, because within the body of the bacteria, this particular cry protein will exist in inactive protoxin form. But inside the insect gut, due to the alkaline medium of the insect gut, this inactive protoxin will be converted into the active form. I'm saying that within the gut of the insect, due to the alkaline medium, the inactive protoxin will be converted into active toxic form, and that will bind to the epithelial cell of the midgut of the insects and will make the surface pore. So there will be multiple pore in the epithelial cells of the gut of the insect, and this will bring soiling and also the lysis of the gut cells, which will lead to the death of the cells. Okay, so most BT toxins genes are insect group specific, and because a number of cry genes code for different cry proteins, so just remember the few of the name of the genes. Okay, so cry1AC and cry2AB gene control the cotton bollworm. This is very important because we're saying that they are insect specific, insect group specific. So first we have got the gene cry1AC and cry2AB gene, which used to control the cotton bollworm, and we have got cry1AB gene, which used to control the corn borer. So these are insect group specific genes. Okay, so biotechnologists, they have isolated the cry gene from Bacillus thuringiensis, and they have introduced these genes into the number of variety of crops to make it pest resistance. And whenever we are going to introduce the gene to the host plants, we require a vector. So in this case, we are using the vector. Those biotechnologists, they were using the vector Ti plasmid of Acrobacterium. Clear? And the transformed crops, they are resistant to the borer caterpillar. And the cry-gene BT gene gets incorporated in the genome of the host cells and express itself, producing the cry protein. So all the processes we have learned in our previous chapter, biotechnology principles and processes. So over here, BT cotton is the first transgenic cotton which has been produced by genetic engineering by introducing a gene from the Bacillus thuringiensis, which produces an insecticidal protein which is toxic in nature, is also called the cry protein. And these cry proteins, they are produced by the cry gene. So kindly go through it. Okay. Next one is the pest resistance tobacco plants, which have been produced by the RNA mediate interference or RNAi. Okay. So plant resistance to nematode pest and the viral infections, they are engineered by silencing gene expression. That means we are going to silence the gene. We are not giving the gene to be expressed. Okay, so here we are trying to suppress the expression of the gene of the pest so that the plant will be free from the nematode pest and from the viral infections. Okay, so this particular silencing is mainly done by the help of complementary double-stranded RNA, which also called the RNA mediate interference or RNA interference, RNAi. And here we'll just introduce the long double-stranded RNA into the cell of the plant. Okay, and mainly the source of this double-stranded RNA, could be from the infection by ribovirus having RNA genome. Clear? So what I mean is that this RNAi, the RNA interference, is one of the natural phenomenon, natural defense mechanism we can say in the eukaryotic cells against the nematodes and the viruses. So here we will learn about one of the examples of the plant which has been developed pest resistance by the help of RNA interference. So we will go through example. Okay, this is very important. So here we have got the example, the nematode parasites Meliodicne incognitia, which will infect the root of the tobacco plant. This is very important. Okay. So the root of the tobacco plant is usually affected by the nematode parasite Meliodicne incognitia. So now we have to develop the tobacco plant which could be resistant from the attack of these nematode parasites. So how to develop the tobacco plant which will have the property of resistance to these parasites? Clear? So for providing the resistance against infection by this Meliodicne incognitia by this nematode, the nematode specific genes are introduced into the host plant. One more time saying that in the tobacco plant we have to introduce the nematode specific gene. Okay, nematode means the Meliodicne incognitia. So we have to introduce the nematode specific gene within the host plant. Host plant is tobacco plant. And for that we will require the vector Acrobacterium. The TI plasmid of the Acrobacterium can be used as a vector for the introduction of nematode specific gene within the tobacco plant. Okay, now these transgenes in the host cells in the tobacco plant, they will produce both the sense RNA and the antisense RNA. And since the sense RNA and the antisense RNA, they are complementary to each other, they will form double-stranded RNA. And this long double-stranded RNA, they are clipped by the RNase enzyme into small short interfering RNA, small interfering RNA, siRNA. Clear? So what we are doing? Within the body of the tobacco plant, we are introducing the nematode specific gene. Now that genes is mainly introduced by the help of the vector Acrobacterium, and within the host it will produce both sense and antisense RNA. And sense and antisense RNA, they have got complementary base pairs, they are complementary to each other, and since they are complementary to each other, they will form double-stranded RNA. It will form a long dsRNA. dsRNA means double-stranded RNA. Now that long double-stranded RNA will be cut into small interfering RNA by the help of the enzyme RNase. Okay. Now this siRNA, small interfering RNA, that molecule will bind to the protein complex called RISC. RISC means RNA induced silencing complex. Okay, so those siRNA which has been clipped or which has been produced by cleaving of the double-stranded RNA by the RNase, now will bind to the protein complex called RNA induced silencing complex or RISC. And this complex, they have got helicase-like activity. Helicase means they will unwind it. They will separate the two strands of RNA. Okay, so this complex has helicase-like activity and it will unwind the two strands of double-stranded RNA. So it will make the double-stranded RNA to unwind and will make it single-stranded RNA. Now its antisense RNA strand, That means dsRNA, it was mainly formed of the sense RNA and the antisense RNA. Now it has been unwinded due to RISC complex. Okay. Now the antisense RNA, it will pair up with the functional RNA molecule of nematode. Once the tobacco plants will be infected by the nematode, the antisense RNA will go and pair up with the functional RNA of the nematode and will cut at its specific sites where the antisense RNA is bound. Okay, so this is called silencing of target mRNA. So what we have done, so once the tobacco plants will be infected by the nematode parasites, mainly or incognitia, we have got already sense RNA and antisense RNA which has been unwinded by RISC. So now that antisense RNA will go and bind with the functional RNA of the nematode and will cut at the sites where the antisense strand is bound. So this will silence the mRNA, which is also called silencing of target mRNA. And as a consequence, the nematode parasites cannot survive in the transgenic tobacco plant. So this is the role of RNA mediate interference or RNAi in the production of pest resistant tobacco plants, because these tobacco plants are being infected by the nematode parasites, mainly incognitia, which will decrease its productivity. So currently go through the viticulture and the pest resistant tobacco plants by RNA mediate interference. Okay. Next one, flavor tomato, is mainly a transgenic variety of tomato which was mainly developed by genetic engineering technique to have the resistance to rotting by using the antisense technology. Normally the tomato, they used to start rotting up by the action of the enzyme polygalacturonase (PG). Okay, that will decrease the peptine. So the ripening can be delayed by blocking or slowing down the production of the enzyme polygalacturonase by the RNA interference. Because this enzyme PG or polygalacturonase, they used to start the process of fruit ripening. So once we will slow down or will block the activity of this enzyme, then obviously the post harvesting loss or the ripening time can be increased. Okay, so this is the example of example of flavor tomato which has been produced to just increase the post harvesting losses or the post harvesting lifespan. So therefore the fruits of flavor tomato remain fresh and retain the flavor much longer time. Okay, then you have a golden rice which is rich in vitamin A. Its grain are yellow and they contain the pigment beta carotenes, and it's mainly the rich source of vitamin A also. And it has been developed by introducing the 2 genes for the enzyme biotin synthase from daffodils and 1 gene from the bacterium with the help of Agrobacterium. Okay, then we have got the engineered brassica napus, the gene coding for the protein hirudin that prevents the blood clotting. We introduced into brassica napus. So the transgenic seeds contain hirudin, which is extracted, purified and used as medicine. So here we have just finished the 6 examples of biotechnology which has been used in the development of genetically modified crop. Okay, so this was the today's class. In the next class, we will begin with a new topic, the transgenic animal. So this much for today. See you in next class. Thank you.